Hi everyone, am I audible to you all? Yes? Am I audible to you all? So hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to talk about the biology and its application. So it is the third session and hopefully the last session of this chapter, and we will be discussing the gene therapy, human insulin, molecular diagnosis, molecular farming, biopiracy, ethical issues. That's it. So this is what we are going to discuss. So tell me everyone, how are you doing? Yes, how are you doing? A very good evening to all of you. Very good evening to all of you. Yes, it is one of the most important chapters for your NEET, right? So that's what we have to cover today. So quickly hit the like button. Hit the like button, and if you're new to our channel, just subscribe our channel quickly. Number of students are more and number of likes are less. So quickly hit the like button and share the video link with your friends as well. And moreover, students, just subscribe to my profile on Unacademy platform, and tomorrow at 10 a.m. right? See, just subscribe to my profile, right? You'll get free classes where I will be teaching you the important topics, and that too is for nothing, right? It's free of cost. You have to pay nothing for that classes. So this is the coupon code that you have to use to join that classes. That's Ambika 10, and free of cost you can learn any topic. So tomorrow at 10 a.m. I am going to discuss the urine formation and counter current mechanism. So just go to the Unacademy platform, subscribe to my profile, and be the part of this special class. Okay, so sharp tomorrow at 10 a.m. I'll be taking the session on urine formation, right? So now let's continue biotechnology and its application. We have already discussed the majority part of this chapter. So today, as I said, we will be starting with students. Yes, so we are going to start with the genetically engineered insulin. Okay, so it is also one of the most important topics. So tell me, what do you know about the insulin? Good evening, Vinod. Good evening, Irfan. Good evening, Subhashin. So tell me, what do you know about the insulin? Happy Diwali. Quick, tell me, what do you know about the insulin? The topic for today is genetically engineered insulin. But before that, do let me know about the insulin. Actually, in this chapter, we are talking about the applications of biotechnology, that how can we use the biotechnology, right? So we know about the flavor saver tomato, we know about the transgenic tobacco plants, we know about the BT cotton as well, we even know about the golden rice. If you know it, very good. Otherwise, note down golden rice also, right? That is also a biofortified product, right? Which word I have used? The word is biofortified. But a biofortification is a process by which the nutritional, right? The nutritional, what is increased, bache? Yes, the nutritional value of nutritional value of food is enhanced. So it is the biofortification, right? And you know that when you are talking about the biotechnology, right? By doing what? By gene, right? By introducing a foreign gene, we can enhance the nutritional value of the food as well. So golden rice is also one example, because golden rice, okay, it is a good source of vitamin A, right? Just a minute. Yeah. So what is it? It is a good source of vitamin A. It is having beta carotene, which is the precursor. Okay, it is having beta carotene, which is the precursor of this vitamin A. So it is very good to treat the night blindness. Persons having night blindness, you know, na? In case of night blindness, what's the issue in night blindness? See, do you know about the pigment rhodopsin? Yes, students, do you know about the pigment rhodopsin? Rhodopsin pigment is present in the rod cells, and rod cells are going to provide the vision in dim light. Okay, our eyes, we are having what? When you talk about the retina, we have the rod cells as well, we have the cone cells as well. So in rod cells, the pigment rhodopsin is present, and for the synthesis of this pigment, vitamin A is very important, because when you are talking about this opsin, it is the protein part, and here, right, retinal is going to make this pigment, retinal. Right, so it is an aldehyde of vitamin A. So vitamin A is very important for the synthesis of this pigment, rhodopsin. So if a person is lacking this, night blindness can occur. So when we have the biofortified food, means the food having the enhanced nutritional value. So one example is golden rice, that is having the gene for the beta carotene, and that beta carotene is the precursor of vitamin A. So that's what you need to remember. Okay, students? Okay, so that's how the biotechnology is used. So today we'll be talking about the insulin, genetically engineered insulin. But before that, you should know what is insulin. But insulin is a protein hormone. What is insulin? Insulin is a protein hormone. Means if you talk about its chemical nature, it is proteinaceous. Right? What is it, Rohit? Rohini? Yes, bache Vinod. What is it, Subhashini? It is proteinaceous. Okay, it is a proteinaceous hormone. It is made up of protein. So, bache, insulin is released by pancreas. So, what is a pancreas? Pancreas is a gland. Right? Pancreas is the second largest gland of our body. The first largest gland is the liver, right? And second is the pancreas. And moreover, when you talk about the pancreas, pancreas is a heterocrine gland. What type of gland it is? It is a heterocrine gland. Means it is a composite gland or a mixed gland. That's what you need to remember. Okay. So pancreas is having exocrine part as well. Pancreas is having endocrine part as well. So when you talk about the endocrine part of the pancreas, here you have certain cells. You have islets of Langerhans, right? You have islets of Langerhans. Clear, bache? And they have different cells. So we have the beta cell here, and that beta cell is going to release the insulin. So insulin is a protein hormone which will be released to decrease the blood sugar level. Like let's say after meal, even in our blood, sugar level means the glucose level is more. So at that time, insulin will be released, right? And the excess of glucose, what that insulin will do, right? Insulin will act on the liver cell. Insulin will act on the fat cells. Liver cells, you know, hepatocytes, right? You know about the hepatocytes, Gautam bache, right? So when you talk about such cells, when insulin is released, so what that insulin will do, students, that insulin will increase the uptake of this glucose by the liver cells and by the fat cells as well. So excess of sugar which is present in the blood, that will be consumed by the liver cell. So basically glucose will be converted to the glycogen. So that's how insulin works. But if there is no insulin, right, if insulin level is low, you know that diabetes can occur. I hope you are aware of it. Diabetes mellitus. Okay, diabetes mellitus. There are two different types of diabetes, na? Insulin dependent, insulin independent. So I'm not discussing that in depth, but I hope you know that insulin is very important. Otherwise, diabetes can occur. And in India, right, there are, you can say that like in every family, you are going to find a diabetic person. Am I right? Right, in every family you are going to find a diabetic person. So insulin is very important. Now, as I said, it is a protein hormone, right, bache? It is a protein hormone. So if it is a protein hormone, means it is having the amino acids. Okay, protein is ultimately made up of amino acids. Many amino acids, they join, they form the proteins, they form the polypeptides. So when you talk about this insulin, it is having two chains, A chain and the B chain. Right, it is having A chain and the B chain. When you talk about the B chain, it is having 31 amino acids, and when you are talking about the A chain, it is having 20 amino acids. That's a very important topic, because insulin is a protein hormone. So of course that protein hormone is having what? That protein hormone is having the amino acids. So it is having A chain as well, it is having B chain as well. A chain is having 20 amino acids, B chain is having 31 amino acids. So overall in an active insulin, okay, right, when you are talking about the structure of an active insulin, total 51 amino acids are present. Right, total 51 amino acids are present. And bache, always remember that insulin is released in inactive form in our body. So before understanding the genetically engineered insulin, you need to know about the basic things related to insulin. Okay? But insulin is itself a protein. Why are you talking about the acid? Amino acids are present, and that amino acids can be acidic, right? But why are you just focused ki why any other acid in insulin? What type of question it is? Rectify that first. Okay, so insulin is released in inactive form. How that insulin is released? It is released in an inactive form. Remember that. And you are going to call that as proinsulin, right? Like in the case of digestive system also, we discussed this thing now, that the proteases, they are released in inactive form. They are not released in active form, they are released in inactive form. So here I'm saying that when you are talking about the insulin, even it is released in inactive form, you call that as proinsulin. Okay, so proinsulin means inactive form. So when you talk about this proinsulin, then remember, it is having 3 chains. A chain, B chain is also there. These are the chains of amino acids, and one C chain is there. Right, one C chain is also there. Clear, bache? C chain is having 33 or 34 amino acid. Right, it can have 31 or 34 amino acids, something like that. So A chain is having 20 amino acids, B chain is having 31 amino acids. This is something very important. You need to remember, MCQ can come from this part. Okay, students? Right, MCQ can come from this part. So if we want our insulin to become active, we need to remove C chain. Right? How can we convert proinsulin to active insulin? We have to remove C chain. Okay, we have to remove C chain. First of all, understand the structure here. Okay, so come to this diagram. Can you see this proinsulin? Can you see this proinsulin? Right, bache? So this is the A peptide here, this is the B peptide here, and this part is the C peptide here. Are you getting it? This part is the C peptide here. Clear, clear, clear? This part. So basically, if we want our insulin to become active, so what we have to do? We have to remove this chain. Fine, we have to remove the C polypeptide. Right, bache? And once it is removed, of course, the insulin will become active. Okay, and one more thing that you need to remember is that one A chain, it is connected to B chain with the help of disulfide linkage. Fine, with the help of disulfide linkage. Fine, bache? Right? See, these amino acids, one amino acid, second, third, fourth, fifth, like whatever amino acids are there. No doubt, they have peptide bonds in between. Right? They are joined together with the help of peptide bond. If you don't know anything related to the peptide bond, do check my biomolecule session. Okay, so they are linked together with the help of peptide bond. And here, bache, this A insulin and B insulin, they are connected together with the help of disulfide linkage. It is another important MCQ. Fine, it is another important. Rohit, bache, it is another important MCQ. Clear? Clear? So now what we need to discuss? We need to talk about the genetically engineered insulin. So insulin is important, that we all know, right, to treat the diabetes. Now students, the point is, like initially, like you can say that many years ago, when genetically engineered insulin was not available, at that time pigs, right, pigs used to slaughter. Okay, first of all, Ramesh, this is not extra content. Okay, this is all related to the NCERT. I think you really need to read the NCERT carefully, Ramesh, first thing. Second thing is that if you don't like it, you can leave the class, simple as that. Okay, Vinod, can't you see disulfide linkage is there, na? Disulfide linkage, 2 disulfide linkages are there. Can't you see this diagram? See this, so disulfide bond is going to join it. Okay, so what am I saying, that when that genetically engineered insulin, when it was not available, at that time pigs used to be slaughtered, okay, which is unethical. And even like when pigs, they were slaughtered, na, so from their pancreas, when they used to take the insulin, okay, so that insulin is not even good for everyone. Sometimes it is allergic, like we are taking insulin from animal body, so it's not mandatory that insulin will provide 100% results. No, sometimes it is allergic to some people. So many issues were there. And above all, the worst thing that I can say is that we are slaughtering another animal so that we we can get a cure, which is unethical, right? So it was very important to have something, a substitute, right, that can treat the diabetes. There comes the, so after biotechnology, there comes the concept of the genetically engineered insulin. It is not just the insulin, right? Biotechnology is used in therapeutics. It is used in different ways, right? These days, like scientists, they are working, they have even worked where they have made the medicines which are further having the coatings. Like, let's say if there is any peptide, now we cannot directly take the insulin. I'll give you one example, right? Now we cannot directly take the insulin. Okay, why? Because it's a protein hormone. So in our stomach, it will get digested. In our stomach, it will get digested. Okay, so because of biotechnology, there are certain, like, oral ingestion of such protein medicines are also possible. How? Because in their case, that peptide bonds will be decreased. Moreover, that protein will be covered with a kind of coating. Okay, so that coating will not allow that protein to get digested in the stomach, and moreover some absorption enhancers are also attached to such medicines, so that immediately they get observed. So biotechnology is helping in this therapeutics in one way or another way. But in the case of insulin, because that in every family you are going to find diabetic people, so that it is important, right? So earlier I told you about the slaughtering of the pigs. Then I'll tell you about the story that how this genetically engineered insulin, it was formed, right, bache? So now let's relate it with the NCERT. So here they have just talked about the diabetes only. Okay, so management of adult onset diabetes is possible by taking insulin at regular time intervals. So obviously insulin is important, right? So as I said, insulin used for diabetes was earlier extracted from pancreas of the slaughtered cattle and the pigs, right? So insulin from an animal source, though, caused some patients to develop allergies or some other reactions are there, because body will recognize that as foreign agent, right, Rohit? Body will recognize it as foreign agent. So when you talk about the insulin, see, the structure is also explained here, right? The structure is also explained here, right? Can you see that, right? That insulin consists of two short polypeptide chain A and chain B, and they are linked together by disulfide bridge, right? Two sulfide bridges are there, so it is connected together by, right, basically disulfide. Di means two sulfide bridges are there. These are the sulfide bridges. Clear, bache? These are the sulfide bridges. So now in the case of mammals, I told you insulin is synthesized as a prohormone. Prohormone means inactive hormone, right? So how can we make it fully matured? By removing the C-peptide. So bache, this part here is important, that main challenge for production of insulin using recombinant DNA technique was getting insulin assembled into a mature form. So obviously it is challenging. Now I'll relate it with the bacteria. Now when you talk about a bacteria, okay, see, don't you think that it is challenging to make the insulin? Don't you think? Because insulin is synthesized from its gene. Let's say if we are taking the gene of the insulin, even from the humans, or if it is artificially synthesized, then what is going to happen? That gene, okay, that gene will form the protein, no doubt. But that protein is that protein is what? That protein is inactive protein. But we want the active insulin. So it is very challenging to form the insulin directly using the gene. So in 1993, right, in 1993, an American company, Eli Lilly, you should know about this company as well. You should know about the story, right? Eli Lilly, they synthesized the insulin by recombinant DNA technology. How? Let me tell you. See, bache, before starting this, I was talking about the bacteria, right? So in the case of bacteria, you know that there is no RNA processing. Do you know that? In the case of bacteria, there is no RNA processing. In the case of bacteria, students, you know that there is no removal of the introns. Do you remember that? There is no removal of the introns. Yeah, sorry, my bad. It is 1983, not 1993. It is 1983. Right, bache? Right, bache? So basically in bacteria, there is no RNA processing. There is no RNA splicing. Do you remember splicing? RNA splicing. So RNA splicing means removal of what? RNA splicing means removal of introns. What are introns? Introns are the non-coding sequences. What are introns, bache Sanvi? What, Rohini, Vinod? What are introns? Introns are the non-coding sequence, right? So that non-coding sequence will be removed and we will get the coded protein, right? Functional protein. But right, in the case of insulin also, if we are directly taking the gene from the humans, or we can say that we are artificially synthesizing that gene, if that gene will be inserted in bacteria, still bacteria will not give us the functional protein. Isn't it? Bacteria cannot give us the functional protein because in bacteria there is no RNA processing, there is no RNA splicing. So obviously it is challenging. So what they have done, they synthesize the A chain and the B chain of this protein. I told you now, A chain is having how many amino acids? 20. B chain 20 or 21? Yes, this is also a point. How many amino acids are there in the A chain? Is it 20 or 21? Yep. Is it 20 or 21? It is 21, of course. 21 amino acid. Maybe, yes. Yes. Here also. Right? Let me rectify this. Let me rectify this part. Yes. So it is the 21. It is 30. So total 51 amino acids are there in functional insulin, but A is having 21, B is having 30. So remember that. Remember that. It's 21 or 30. I'm rectifying this. You also please note down. Okay, you also please note down. Actually, see, I just forgot my trick. So that's why by mistake I mentioned 20. See, A means first alphabet, first letter. A means what? A means first letter. So that's why I remember that 1 is there in the A chain. Right? This is my way to remember this. That A is the first letter. A is the first letter. So 1 is there in the A chain. So that's why it is 21. And when you talk about B, B is like this. Right? So means it will be having 30 amino acids. So in totality, it is 51. Okay. In totality, it is 51. Clear, bache? Right? Understood? Fine. So A chain is having 21 amino acid. B chain is having 30 amino acid. So in totality, we have 51 amino acids. So they synthesized this A chain and the B chain separately and later they joined it. So it was exactly like the human insulin. And which bacteria they have used? E. coli. Another important MCQ. Right? E. coli. Clear, bache? Clear, bache? Ready, bache? I cannot help it because whatever you are thinking, it is not appropriate. Right? Once I'll see the messages, obviously I will comment. Okay, so kindly focus here on this, right? On whatever I'm explaining, you will get the answer of all your doubts. Done, bache? So in 1983, Eli Lilly, an American company, see, they prepared two DNA sequences corresponding to A and B chain. Okay, bache? And then they introduced them in a plasmid of E. coli to produce the insulin chain. So by combining chain A and B, right, bache, they created that human insulin. Fine. They created that human insulin, and obviously it was called as humulin. What is it? This genetically engineered insulin is humulin. So it's ditto like the human insulin. So no side effects are there. So no slaughtering of pigs, right? And obviously ethically it is also right in that regard. Fine. Done, bache? Okay. Done. So it is always in pure form. So there is no chance of contamination, no slaughtering of animal is there, no immune response, no side effects are there, right, bache? So we have even the sufficient manufacturing capacity so we can get it at a high, on a large scale also. Fine. So this is about the human insulin. So next is the gene therapy. So do you know anything related to the gene therapy? Do you know what is gene therapy? Anyone? What is gene therapy? Anyone? Bache, gene therapy. Bache, acha, Uddhav is saying, ma'am, how did they use the E. coli? Because it cannot form RNA or DNA. Who told you, bache? In the E. coli's plasmid, this gene for, like, let's say they will introduce a gene which can be synthesized artificially also, that will form the A chain only. Simple. Simple. Okay. So that will that is what they are going to introduce, and this is how it will help. Okay, so next point is the gene therapy. Gene therapy what? But it is the collection of methods. In gene therapy, we are not talking about only one technique. It is that which is used to alter a defective gene. Let's say if in my system there is any gene which is defective. So defective gene will give me the defective protein. Means there will be some defect in my metabolism. What if that protein, right? What if that protein is playing a very important role in metabolism? Obviously, my metabolism will be affected, right? Any disease can occur, any disorder can occur, right? So basically, when we talk about the gene therapy, as I said, it's a collection of methods which is used to alter a defective gene, which is used to change a faulty gene with the correct one. Okay, with the correct one. Are you getting it? That is what gene therapy is. So see, if a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Of course, yes. So if there is any hereditary disease, means there is some issue with some protein. So it's and that too because of the gene. So that can be rectified. But here we need to understand one thing. Now let's relate it with the development part. So when you talk about the zygote, you know that we all are developed from a single cell that is known as zygote, right? We all are developed from a single cell that is known as zygote. You know that zygote will divide. There are many stages. The morula will come, the blastula will come, blastocyst will come, implantation will occur. This is what we need. This is what we know, yes or no? Zygote is there, it is single cell, right? We all are developed from the single cell, and you know that in humans also after 30 hours, the zygote will take its first division, right? So basically, zygote will divide into two cells in 30 hours, right? It is the slowest. Actually, the mammalian cleavage is the slowest cleavage of the animal kingdom. If you know it, very good. Otherwise, note down this point that mammalian cleavage, it is the slowest cleavage of the animal kingdom. So your zygote will divide. It will form two cells. It will have 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage. You know it, right? So initially, when the zygote is dividing, initially when division is there, right, the cells, they're just dividing randomly. Their fate is not fixed. Fate is not fixed means they don't know what they have to form. Okay, they just know one thing, that we have to divide. So basically, initially the differentiation is not there. You know, na? Firstly, differentiation will occur and then maturation will occur. Differentiation will occur and then maturation will occur. I hope you are aware of it. So initially what is going to happen? The differentiation is not there. So at that time, you know, there are many techniques. Okay, by using such techniques, right, if it is detected that there is a genetic disorder in that fetus, so obviously then it will be rectified. Okay, gene therapy will be used, and by this gene therapy, what is going to happen? The faulty gene will be replaced with the corrected one. Then it can be cured for the lifetime. Okay, then it can be cured for the lifetime. So basically, in embryonic stages, if we recognize any genetic defect, if we recognize such defect, right, then it can be rectified. But later on, because all the cells are differentiated, they know what they have to do. They have a specific role. In that case, it is not, even the gene therapy is not going to provide the permanent solution. Clear, Subhashini? Even in that case also, gene therapy is not going to provide the permanent solution. So it is written here as well. So here you people can see that gene therapy is an attempt to do this, to rectify this. So gene therapy, as I said, is a collection of methods that allow the correction of gene defect that has been diagnosed in a child or in an embryo. So genes are inserted into a person's cell and tissue to treat that disease. So correction of genetic defect involves delivery of normal gene into the individual or embryo to take over the function and to compensate. So when you talk about the gene therapy, we have one classical example, right? We have one classical example. So in 1990, right? So gene therapy, for the first time, it was used for a 4-year-old girl. Okay, and that girl was having the ADA deficiency, right? The gene for ADA enzyme was defected then. Okay, the gene for ADA enzyme was not present, it was defected. So first of all, understand what is the meaning of this ADA. ADA means, bache, adenosine deaminase. What is the meaning of ADA? ADA means adenosine. It is adenosine deaminase. Clear बच्चे? बच्चे, it is an enzyme. It is an enzyme, right, which is used for the formation of that lymphocytes. I hope you know about the lymphocytes. Do you know about the lymphocytes? What are lymphocytes? What are lymphocytes? They are the WBCs, right? They are the white blood cells. And you know that we have B lymphocytes as well, T lymphocytes as well, right? What do we have? What do we have? We have the lymphocytes. B lymphocytes are also there. T lymphocytes are also there. So B lymphocytes will make your antibody mediated immune system. They both are playing role in the immune system. T cell will make your cell mediated immune system, right? T cell will make what? T cell will make your cell mediated immune system, right bache? Yes or no? Yes or no? Right. So basically this enzyme ADA, it is very crucial for immune system to work. Okay, it is very crucial for immune system to work because this enzyme will form these lymphocytes. This enzyme is not there in our body. We do not have the lymphocytes. No lymphocytes means no immune system. Neither the cell mediated immune system will work nor the antibody mediated immune system will work. Are you getting it? Are you getting it? So gene therapy, for the first time it was used for a girl, for a 4-year-old girl in 1990, and she was deficient with this particular enzyme. So if this enzyme is not there, immune system is not working, means that girl is having the disease which is known as SCID. So what is SCID? SCID is severe immunodeficiency syndrome. What is SCID, students? It is severe immunodeficiency syndrome. Again, repeat it. What is SCID? It is severe immunodeficiency syndrome. Severe combined immunodeficiency syndrome. So in the SCID, like, it's a very rare disease. Of course, autosomal recessive it is. So it's very rare. Okay, so in that case, that person is deficient with the ADA enzyme. So if person is deficient with the ADA enzyme, means the immune system is not functional. So in that case, that baby will not have the long life, right? Even the body is not having the capacity to fight with the very basic disease, like not to fight with the fever, with the common cold. Are you getting it? So it is severe combined immunodeficiency syndrome, right, bache? That is your SCID. Okay, so in that case, immune system is not functional at all. So when you talk about this particular case, right? When you talk about this particular case, so here what was done, bache? What was done? Yes. So here in some children, that ADA deficiency, it is cured with BMT, or one is ERT. BMT means bone marrow transplantation. Bone marrow transplantation. ERT means enzyme replacement therapy, right? Second is what? It is enzyme replacement therapy, and BMT is what, bache? It is the bone marrow transplantation. So actually here what was happening, that you know that this enzyme will form in the site of production of enzyme is the site of production of this ADA enzyme is your lymphocyte. So in gene therapy, because I mentioned it, it's a collection of techniques, right? So BMT and ERT is performed. So even BMT, the bone marrow transplantation, even it is not 100%. It will not provide cure 100%, right? Not even the enzyme replacement therapy. Okay, both are not providing the 100% cure. Clear, bache? As I said, if in embryonic stages these defects they are recognized, then only, right? Then only the 100% results will be seen. The disease will be 100% cured. Otherwise, there is no chance. Okay, otherwise there is no chance. And in this case, what was done, bache? In this case, what was done? The enzyme replacement therapy was used. Clear, bache? So as the first step of this disease, as the first step of this treatment, what was done? The lymphocytes were taken from the patient's body, right? The lymphocytes were taken from patient's body, like blood samples were collected, no? So lymphocytes were taken from patient's body, right, bache? Because here what was done, bache? Ultimately in the lymphocytes, this ADA enzyme was inserted, right? They didn't change that faulty gene because it was recognized later. So what they did, they just replaced the enzyme, because ultimately ADA was deficient. So see, See this part, that first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Very important point, bache. Definitely MCQ will come from this part. Okay, then come to this point, right? See, in some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, right? So in which functional ADA is given to the patient by injection. So functional adenosine deaminase will be injected. So what will be done? First of all, blood will be taken, right? Let me explain it, then let's relate it with NCERT. So when blood from that patient will be taken, na bache, so in the lab, right, there in that lymphocytes, what will be done? Ultimately, lymphocytes are deficient. They do not have the ADA. The site of production is what? The site of production of ADA is what? Lymphocytes. So no enzyme means no proliferation, means no immunity. Then when lymphocytes outside the body, when they will be grown in the lab, no? So with the help of a suitable vector, that functional gene will be introduced in that enzyme. And here, which functional gene will, which vector will be used? Retrovirus. Right, retrovirus, because you know that normally when we talk about the viruses, naturally it is a part of virus, it is a part of virus life cycle, right? That virus, it is going to use the host's machinery. What virus will do? Virus will use the host machinery for its replication. Virus is going to insert its genome into the host genome. So here in this technique, what was done? A retrovirus was used. Retroviral vector was used. And you know how to use such vectors. You know that this retrovirus means the virus having RNA as a genetic material. So you know that this virus will insert its genome into the host's DNA. Okay, so in the case of this retroviral vector, what will be done? Bache, its harmful genes, they were disarmed, right? Their harmful genes were disarmed. Okay, and then this viral vector will be used to introduce our gene of interest, and here our own gene of interest will be the gene that will synthesize ADA, that is your adenosine deaminase enzyme. So here you people can see, just look at this. Okay, just look at this part. So in others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. So but the problem with both of these approaches, that they are not completely curative. So as the first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. Then but you see a functional ADA cDNA. So what is this cDNA? cDNA means complementary DNA, right? But disarm means, Vishnu, basically the harmful genes will be removed, right? So that virus cannot introduce the harmful genes in our body. Okay. Okay, so as I said, see, what is the word cDNA? cDNA means complementary DNA. What is the meaning of C here? C means complementary, right? C means complementary. So it is the cDNA, complementary DNA. Okay, you know na reverse transcription, you know na, like even it is a retrovirus. So retrovirus means it is having RNA as its genetic material. What is the meaning of retrovirus? It is having RNA as its genetic material. So from RNA, DNA will be formed with the help of enzyme reverse transcriptase. With the help of enzyme reverse transcriptase. So this RNA will form the DNA, that will be the single stranded DNA first. So this is the complementary DNA. It is formed from the RNA. So that's why here it is written, bache, that a functional adenosine deaminase, right, complementary DNA using a retroviral vector is then introduced into these lymphocytes, and then these lymphocytes will be re-injected in the patient's body, right? That lymphocytes will be re-injected in the patient's body. Are you getting it? Are you getting it? Yes, bache. Bache, complementary to that RNA, na, Vinod. Bache, what is the reverse transcription? From mRNA, DNA will be formed. From mRNA, DNA will be formed. So that DNA is complementary DNA. C means complementary. And obviously, that DNA is synthesized complementary to the mRNA. This is simple. You know that here the rule of complementarity work. Okay, So this is what you need to remember. So now the lymphocytes, they will be returned to the patient's body. Now that patient is having what? ADA enzyme, means immune system is functional. But the problem here is that lymphocytes are mortal. They are not immortal. They will not survive for the lifetime. So this process has to be repeated again and again, again and again, again and again. And because of that, what is going to happen, student? Right? You know that it is going to be physically draining. It is going to be financially draining. Isn't it? Isn't it? Yes or no? Isn't it? That is draining, right? Right? So this process has to be repeated again and again, again and again. So if a person having SCID, there are very less chances of their, yes students, there are very less chances of their survival. Very less chances are there. Got it? So this is about the gene therapy. Understood? This is about the gene therapy. So understood, bache? Sure? Sure? Are you sure? Any other doubt? Subhashini, Kevin? Okay. Okay, so next thing is the molecular diagnosis. So tell me, what do you know about the molecular diagnosis? Next topic is molecular diagnosis. Yes, next topic is molecular diagnosis. So what do you know about that? What do you know about the molecular diagnosis? Even it is very easy. Even it is very easy. See, normally whenever you talk about any of the disease, like let's say the example, let's take the example of typhoid, right? For the typhoid, do you know which one is the test that need to be performed to diagnose whether there is the typhoid or not? Can you name the test? Basically, I'm talking about the blood examination. Blood test, basically, yeah, I'm talking about the blood test. Exactly, Subhashini. Very good, Sanvi. Very good, Subhashini. Okay, that is the Vidal test. है ना Vishnu? Vinod, that is the Vidal test. है ना? That is the blood test. Okay. So now the point is, when do we get to know about such disease? Like, what is the basic procedure? You know that in general what we used to do. If we are not feeling well, first of all, we'll try the home remedies. It's a fact or not? If we are not feeling well, the very first thing that we are going to do is the home remedies. We will go for the home remedies. After that, okay, if still we are not feeling well, then we will start taking the medicines. Okay, and the famous one is the Dolo. After COVID, you know that the famous one is the Dolo. And still if we are not feeling better, then we will think that now we should visit the doctor. Right? That's the reality. So basically, whenever we go for the whenever we visit the doctor, what they used to do, the conventional ways are there, right? The blood test, the urine test, basically body fluid test will be done. Okay, these are the conventional ways, the traditional ways, that in general go for the blood test, go for the urine test, go for this sample, go for that sample. But in these samples, when do we see the infection? When are we going to see the antigen? Right? If you're talking about the blood, if you're talking about the urine, right, when will you see the antigen here, or you can say that the pathogen, yeah, whatever it is, right? Basically, when do we see, when are we going to figure out that, yeah, there is a problem? When that pathogen has, right, when pathogen has caused the disease, when that pathogen has hijacked our body. That's a fact, right? That's a fact, right? Of course. So in the conventional ways, that is the problem, that we will only diagnose the disease when that pathogen is present in our body, when that pathogen has hijacked our body. But when you talk about the another technique, so another techniques means, what are we talking here? Right? I'm saying na another technique. So another technique means, bache, right, rDNA technology, PCR, ELISA. Another techniques means what? It means rDNA technology, polymerase chain reaction, and ELISA, that is your enzyme linked immunosorbent assay. So they are going to, these are the techniques which will help in the early diagnosis of the disease, right? How are they going to help? They are going to help in the early diagnosis of the disease. So blood test, urine test, they will never ever they will help. They are never going to help in the they are never going to help in the in what, in early diagnosis, because they will only will show you the pathogen, the antigen, or anything right, that can tell us that there is a problem when that pathogen has already damaged our system, when it has already affected our pathology, affected our physiology. But here in this case, what is going to happen, students? The early diagnosis will help. So when you are talking about the PCR, you know what is the meaning of PCR, right? That is your polymerase chain reaction. Okay, bache, and this technique is used for the amplification. Why this PCR is used? PCR is used for the amplification of the gene. If you have a gene in very, if you have the DNA available in very less amount and you want to read that DNA, to study that DNA, obviously you can make multiple copies of that DNA by using the technique PCR. And I hope, just for your quick revision, I hope you remember PCR means dye, right? Dye. These are the 3 steps here, right? This is the sequence of that 3 steps. PCR, polymerase chain reaction, dye, denaturation, annealing, extension, denaturation, annealing, extension, denaturation, annealing, extension. Okay, so it is used to make multiple copies. Now if you are thinking that how that PCR will help us, let's say if in our body, if in any of our body fluid, even if there is a very less amount of that antigen, if there is a very less amount of that pathogen's DNA, okay, if there is very less amount of that pathogen's DNA, so by using PCR, that pathogen's DNA can be amplified. Yes, Kevin, that can be amplified. Obviously it can be amplified. So when it is amplified, it can be studied. Okay, so that's how the PCR is used. And for early detection of HIV, that is your human immunovirus, right? For early detection of your HIV, that is your human immunovirus, this PCR is used. Getting it? This PCR is used. Yes, bache? Yes, bache? Okay, done. So that's how this PCR is going to help. Clear, bache? So it is used to detect HIV in suspected AIDS patient, and even it is used to detect the, right, it is used to detect HIV in suspected AIDS patient, even it is used to detect if there is any gene mutation that can, right, in that case also it is going to help. And then comes the ELISA. What is that ELISA? It is the enzyme, right? It is the enzyme linked immunosorbent assay. It is enzyme linked immunosorbent assay. That's what ELISA is. Clear, bache? That's what ELISA is. Enzyme linked immunosorbent assay. Bache, now this technique, it is based on, again, it also help in early detection. Even if pathogen is in very less amount in our body, still it can detect that, means that antigen. Okay, so it is based on antigen antibody interaction. It is based on what? It is based on antigen antibody interaction. Let me explain how. Let's say if a patient, if it is suspected for a particular disease, even if that antigen is present in very less amount in my blood, that can be detected with the help of ELISA. See, if let's say if in patient, just a minute. So let's take the example of Chintu, right? Let's take the example of Chintu. So Chintu is having some issue, right? Chintu, let's say any antigen is there in Chintu's body, right? Even if that antigen is in very less amount, don't you think the body, once body will recognize it, body is going to make the antibodies against it? Yes or no? Yes or no? Yes, Anvi? Yes or no? So even if Chintu's body is having antigen, even if it is in very less amount, so obviously, right, body is going to form the antibody against it. Even if it is in very less amount, it is, once body will recognize it, body will make the antibodies against it. So now let's say the patient's blood is taken. So if in that blood, we can go for two things. We can go for the antigen detection also. We can even go for, if we are suspecting something, no, we are suspecting a disease, we can even go for the antibody. Right, we can even go for checking the antibody. So when you are talking about this ELISA, right bache, it is mainly used for the detection of viruses, fungi, bacteria, right, it is used for detection of viruses, fungi, bacteria, even the mycoplasmas, etc. Right, it is used to detect all that things. Right, it is used to detect all that organisms and quantitate very small amount of antigen, antibody, or even a, right, even a protein. Are you getting it? Or even a protein. Are you getting it? Yes, bache. Are you getting it? So now let's say I want to check one antigen. Right, I am suspecting something and I'm thinking that that antigen is there in Chintu's body, and I'm going to check that with the help of ELISA. So obviously we should have the antibody, right? We should have the antibody against this antigen. We must have that antibody that can bind with this antigen if we want to perform the ELISA. Right, what should we have? We should have the antibody that can bind with this antigen. So the sample will be taken, there will be the trace, right, there will be the kind of trace, the ELISA trace will be there, right, that ELISA trace will be there. Like let's say if this is the tray, so in that tray small wells are going to be there. Right, I'm not explaining you this process in detail, I'm just giving you the overview of this process. Always remember, this process is basically dependent on the antigen antibody interaction. Right, it depends upon the antigen antibody interaction. So basically the ELISA tray or the Petri tray is there. So that tray is having the wells, the spaces where we are going to load the sample. So we must have the antibody against the antigen that we want to detect. So let's put the sample there, right, that fluid sample there, and then the antibody complementary to that. So you know that if there is any antibody which is complementary to this antigen, of course it is going to find that. Right, you are taking the body fluid sample, you are putting that antibody, right? So what is going to happen? The antigen and antibody, they are going to join, yes or no? Antigen and antibody, they are going to join, they are complementary to each other, they will join. So again after that washing will be done, the unbinded antibodies, they will be removed. Okay, even if, I'm repeating this again, even if that antigen is present in very less amount, you are putting the antibodies in that trace, right? Basically, first of all, when you will put the antigen, when you will put the body fluid here, we need to make our antigen immobile. I hope you understand the meaning. We have to make the antigen immobile, right? It should be immobilized against the wall of this well. It should be immobilized against the walls of this particular well, right? Why? Because if it will be fixed, so whenever we will put the antibody, that antibody will join. Okay, that antibody will join. Clear, bache? Clear, bache? Yes or no? Yes or no? Right, so the first thing is antigen, we need to immobilize that antigen, then put the antibodies, they will join, and then whatever antibodies are, you know, left, they are not joining with any antigen, with the when we will done the washing, it will be removed. Then bache, see, there is one antigen, so everything is happening on this well surface. There is one antigen which is immobilized, so antibody will join to it, right? Now we will add another antibody, right? After it, what will we do? We will add another antibody, right? What are we going to do, students, here? We will add another antibody, and that antibody is going to, it is complementary to this antibody. But what is the speciality about this about the second, or you can say that the secondary antibody, that it is attached to a particular substrate as well. Right, it is attached to a particular substrate as well. So this antibody, when it will join here, the substrate is also present. So this substrate is kind of chromogenic, it is the color producing, right? It is what? It is chromogenic. It is the color producing. So actually the second antibody that we have added, no, we call it as antibody, and you know that antibody is also known as immunoglobulin. So you can also call it as anti-immunoglobulin. What is it? It is also known as anti-immunoglobulin. Again, I'm repeating this. You have a tray, right? You have a ELISA tray. In that tray there are the wells. You are loading your sample there. In that sample antigens are present, right? Antigen will be immobilized, of course, with the help of chemicals. Then antibody complementary to it will be added. Then another antibody, that is anti-antibody, or you can say that anti-immunoglobulin, right? Anti-immunoglobulin, it is allowed, it is added and allowed to react, right? Allowed to react. Clear, bache? So this antibody is linked to a suitable, it's linked to a proper enzyme, okay, like peroxidase. Fine, like peroxidase. I'm giving you one example. It will be attached to what? It will be attached to an enzyme like peroxidase, right, bache? And unreacted antibodies will again be washed away. So substrate of this enzyme, a suitable reagent will be added. I told you initially this is the substrate. No, this is the enzyme, right? So I am giving you one example that can be peroxidase. So now the substrate will be added here, right? So if this enzyme is attached to, if this antibody is attached to this one, right, to this complex, so obviously the color will be produced. In the ELISA test, what is going to happen? What is going to happen? The color will be produced, and that color, the intensity of that color will tell us about the antigen. Okay. Okay. So substrate of the enzyme is added, bache, along with the necessary reagents. So activity of the enzyme is going to give us the colored product, and its intensity is going to tell us the amount of antigen. If it is the color's intensity is more, means in the blood the antigen is more. If it is less, then it will tell us less antigen is there. But even if antigen is present in very minute quantity, we'll get to know about it. Clear, bache? We will get to know about it. So that's how this ELISA helps. So it is highly sensitive. It can detect antigen even if it is in the range of nanograms. Imagine. And we cannot do it in our conventional test, right? Even if it is in the range of nanograms. Okay, even if it is in the range of nanograms. Getting it? Yes. Are you getting it? Sure. So it is also used to check, bache, AIDS or other STDs, you know na? And moreover, for confirmation of pregnancy, this ELISA is performed. The amount of HCG, human chorionic gonadotropin, in blood is checked. Okay, the HCG, human chorionic gonadotropin, in the blood is checked. Okay, so other than that, hepatitis, phenylketonuria, tuberculosis, they are also detected by using this ELISA. Okay, they are also detected by using this ELISA. Clear, bache? So enzymes used are peroxidase, it can be alkaline, phosphatase also, right? It can be alkaline, phosphatase also. Fine. So there are many enzymes that can be used here, right? So this is important. Okay, this is important. Done, bache? Sure. Done, bache? Okay. And moreover, bache, some probes are also used. That probes can include the DNA or RNA probes. Let's say ki we are expecting a gene got mutated, that's why the disorder is there. So we can, insert a gene or we can just clone a cell, right? In that cell or in that cell we can study that, we can check whether that gene is, mutated or not. So DNA probe or RNA probe can be used. So when you are using the word probe, so that DNA or RNA sequence, it will be having the radioactive compounds. So radioactive tagging will be there so that we can check, right? So if you are using DNA or RNA probe, let's say you are, suspecting something, there is a prognosis of something, or we have to check. So obviously let's say if there is a disease because of the change in the gene sequence, and we know that particular sequence, so obviously complementary to it we can add a probe if it will get hybridized, right? We'll get to know about it. If not, then again we'll get the result. Okay, so when it is a probe, it is a single stranded DNA or RNA tagged with the radioactive molecule, allowed to hybridize with the complementary DNA, with the cDNA in a clone, right? In a clone cell, it will be allowed to hybridize. Clear, bache? And then it will be detected using radioiotography. Okay, okay. So if we are suspecting that mutation is there, and if we are putting that DNA or RNA probe complementary to this cDNA, if we are putting it complementary to this cDNA, then of course, if it is mutated, it will not hybridize, we will not get the band. So that is also another way. Fine, that is also another way. So this is about the molecular diagnosis. What is it? It is about the molecular diagnosis. Got it? Got it? So that's how the biotechnology, the recombinant technology is going to help. Understood, bache? Done. So next topic is of vaccines. Fine, we also discuss about the vaccines in the chapter Human Health and Disease, but here we will be talking about the DNA vaccine specifically, right? What are we going to discuss here, bache? We are going to talk about the DNA vaccine. What is your homework? You will tell me. Chalo, I'll tell you. Let it be. So DNA vaccines, they are made against the infections like herpes virus, against hepatitis B, right? Against hepatitis B virus. So which vaccines are used? DNA vaccines. So do you know anything related to DNA vaccines? Yes. Do you know anything related to DNA vaccines? Alvin, ma'am, can I write the NEET even if I'm not a biology student? No, bache, you cannot. Biology is important for NEET examination. So tell me, yes, students, tell me about it. Quick. Tell me about it. Tell me about the DNA vaccine. Whatever you know about this vaccine, just let me know about it. Just let me know. DNA vaccine. But without biology, you cannot, Alvin. Biology should be there. Even if it is additional, I think then also it is allowed. What do you know about the DNA vaccine? Yes, students. Let's say, here what will be done? Let's say there is any pathogen, right? We are going to take that pathogen's genes. Okay, let's say there is any microbes. So microbes antigen gene are taken. Microbes antigen, like let's say if there is any pathogen, of course that pathogen is going to cause the disease. There must be some antigen that can be detected by the body. So what are we going to do in DNA vaccines? That antigen will be taken, right? What will be done, bache? That microbes antigen, the gene for that microbes antigen will be taken. I'm repeating this again, right? I'm repeating this again, bache. What will be done? Yes, tell me, what will be done? What will be done? Genes for a microbes antigen, right? What will be done here in DNA vaccines? The gene for microbes antigen will be introduced in the body, right? Gene for, yes, gene for microbes antigen will be introduced in the body. Okay, then some cells of body are going to take up this DNA. Yes or no? Some cells of body, they are going to take up this DNA. And if they will take up the DNA, what will they do? They will obviously, because that gene is for the microbes antigen, so obviously that cells, I'm not saying that all the cells, some cells will take up that DNA and they will start making, because they have incorporated that DNA, they will start making the antigen of that DNA. They will start making the antigen of that microbe. Yes or no? They will start making the antigen of that microbe. Yes or no? Yes or no? Of course. So in that case, what is going to happen? Certain cells of the body, they will start showing that antigen on their surface, right? They will start showing that antigen on their surface. So if they will start showing that antigen on the cell surface. So obviously that will be detected by the antigen presenting cell of our body. Our body will start making an immune response against it. Yes or no? So actually these DNA vaccines, obviously it will be done with the help of this biotechnology, of course. So here it is a kind of mechanism where, you know, our body itself is making the antibodies against a particular antigen. So here, what is the different thing that we are doing? We are just taking the gene of that microbe's antigen that will be introduced in our cells. Some cells will take it, they will make that antigen will be expressed on the cell, and body's immune system is going to recognize that. Yes, bache, body's own cell, they will become the vaccine making factory. They will create the antigen. That antigens will, right, they will create the antigen. Our body cells, they will also become that, you know, that vaccine forming factory. So what will be done? That antigens, they will create the immune response. And always remember, these antigens cannot cause any harm to our body. They are just sufficient enough to create the immune response. And after that, what is going to happen, students, after that, what will happen? Our body's immune response will become, right, our body's immune response will become active, and we will have the immunity. So that is how this DNA vaccines, they work. Clear, bache? Clear, bache? So we so we do not have a full microbe, so it is not going to cause disease in our body. We just have few genes of that microbe that are sufficient to create the immune response only. Fine, bache? That is sufficient to create what? That is sufficient to create the immune response only. So this is about the DNA vaccine. So this is also the application of what? The application of your biotechnology. Okay. So next thing that we need to discuss is, bache, transgenic animals. Can you give me the example of transgenic animals? Everything is given in NCERT, and yes, you have to revise NCERT. Right? Can you just give me the example of transgenic animals? Yes, students. Can you just give me the example of transgenic animals? But please stop chatting, otherwise I have to end the class. Right, Kevin, Rohini, I know you people are here to waste your time. Okay, but I really don't have time for all this. You please stop chatting, or else I'll end the session. Ma'am, for the transfer of any desired gene to any animal vector, what will be the host to transfer, like TI plasmid, such that? Priyadarshini, I really don't understand what are you trying to ask. If you are asking that in the case of animal, how can we insert a gene? Okay, there are many ways, bache. I think you don't remember that other techniques also. The gene gun is there, the ballistic method, okay, the electroporation, the chemical method. So there are different ways. Okay, there are such different ways. So transgenic animal, this is what we have to study. So transgenic animals, even transgenic microbes. So let's start with the animals first, then we will talk about the transgenic microbes. So bache, whenever the word is transgenic, okay, obviously it is genetically modified. Whenever the word is transgenic, of course it is genetically modified. Transgenic means it is having a foreign gene, right? It is having a foreign gene. Foreign gene means gene from some another species, of course, right? Right, so we have many examples of the transgenic animals these days. And if you talk about the first transgenic animal, right, first transgenic animal produced was the super mouse. Do you know that? Do you know that we have many transgenic animals, like transgenic mouse, transgenic pig, etc. There are many, and the first transgenic animal formed was a super mouse. Why super mouse? Because it is having the gene. Which gene? Having a foreign gene. Which gene? Gene having human growth factor. Right? It is having human growth factor gene. So obviously that's why it is a super mouse, because the growth will be more in that case. Right? The growth will be more in that case. So this is a transgenic animal. So if I talk about the transgenic animals, how are they beneficial? Right? How can they help? These transgenic animals, how can they help? It's very simple, students. We can use such animals for the study of a disease. How come the study of a disease? Let's say there is a disease, right? Scientists, they don't know the life cycle of a particular microbe or the pathogen. They don't know how that disease is affecting, whether it is affecting the liver first, this first, that first. So in that case, they can introduce, let's say if it is a microbe, they can introduce that in such transgenic animals. Then they can study that, right, how that they can study the pattern of that disease, that how that disease is growing, one way. Secondly, they can also study any physiological process, we can no doubt for the studies we can do. We can even use these transgenic animals to study the action of a drug, that if we are inserting a drug, how that drug is acting in that, you know, that transgenic animal's body. Moreover, we can study any physiological process in our body. Okay, if that physiological process will be known, if there is any you know, defect in the metabolism, or if there is any change from that physiological process, we can, you know, note down that if there is any change, this change can lead to this disease and that disease. Are you getting it? So that's how it works, Rohini. That's how it works, transgenic animals. Okay, okay. And not just for this, even for studying pathophysiology. Pathophysiology means physiology, you know, normal functioning. Pathophysiology means when it is defected. So even the role of certain genes can be studied. Like we don't know about a gene, that how that gene is what that gene is doing in our body. So that gene can be introduced in a transgenic animal, and we can monitor, right, what is it doing. The role of growth factors can be studied, the role of another genes can be studied. So in different ways these transgenic animals are used, and moreover they can be used as a factory, right? Like we call it as molecular farming. Let me explain this part also. Let's say if in an animal, right, in milching animals, if we are introducing a gene, right, and that gene is providing us a certain a vitamin or any particular protein or anything which is nutritional for us. So let's say if I'm introducing a particular gene in a cow, okay, in a cow I'm introducing the gene that I want a alpha-1 antitrypsin. Just giving you one example, right, that I want the alpha-1 antitrypsin, and that gene will be inserted in a cow. So obviously in that cow's milk you are going to get that product, right? You are going to get that product that will be healthy, isn't it? That will be used to treat the disease. So in that way we can make these transgenic animals like the factories by which we can get the products that we want. Obviously it is unethical, but I'm just telling you that how scientists they use it. So it is called as molecular farming, when these, you know, plants, these animals, they are used to synthesize certain chemicals. Okay, when they are used to synthesize certain chemicals. Are you getting it? Are you getting it? So basically, so transgenic animals which are having foreign genes, right? First transgenic animal was super mouse, and 99% of all existing transgenic animals, they are mice. Imagine 95% of all transgenic animals. And why do we make them transgenic? Like sometimes, you know, sometimes we have introduced the foreign genes. Sometimes, you know, in some transgenic animals, their lifespan is less, right, and they are producing more offspring. So that can also be used to study a disease, to study, you know, any pattern of a disease, to study the effect of a drug. Are you getting it? So there are many examples. Let's take the example of the biological products that we can get. Okay, biological. So I gave you the example of the milch animals as well. So if I have to give you a proper example, as I said, the important biological product being harvested by this method is alpha-1 antitrypsin. What is it? It is alpha-1. I will just write down the product name, alpha-1 antitrypsin. Do you know about this protein, bache? Bache, it is used to treat emphysema. Do you know what is emphysema? Emphysema is caused because of the cigarette smoking. Those who are the chain smokers, they are going to get this emphysema. You know, na, lungs, the exchange will also get affected, right? In the case of lungs, what is going to happen? Alveoli. In the case of alveoli, what is going to happen? The elasticity will not be there. The exchange, the gaseous exchange will be affected. Okay. So to treat emphysema, alpha-1 antitrypsin is required. The word is antitrypsin, right? It is going to, of course, it is going to damage the trypsin, right? So it is alpha-1 antitrypsin. It is used to treat emphysema. So we can use milch animals, bache. We can use that milchy animals, right? That genes can be introduced in their body from their milk. In their milk, obviously this alpha-1 antitrypsin will be present. So this is important example. And what is your homework? Right, you have to mention the answer in the comment section, bache. You will tell me that how this alpha-1 antitrypsin, it treats emphysema. That's what you have to tell me. Okay, that how this alpha-1 antitrypsin, it treats emphysema. That's what you have to tell me. Okay, that's what you have to tell me. Then comes the tissue plasminogen activator. Okay, these animals can be used for making this tissue plasminogen activators. Okay, and other than these tissue plasminogen activators, blood clotting factors. Fine, blood clotting factors, right? That can also be obtained from these animals, right? Even for the phenylketonuria and for the cystic fibrosis also. Fine, bache, cystic fibrosis also. Okay. Okay. Done? Clear? So this is one example. So for the biological products, for important biological products, the transgenic animals can be used. Now let's talk about the first transgenic cow. That is, you all were taking the name. So first transgenic cow, that first transgenic cow is Rosie, right? Rosie. And what was special about this cow, bache? Right, so in 1997, this Rosie cow, it was transgenic cow, it was the first transgenic cow. It was basically formed in 1997. Okay, 1997. So it yielded milk with high protein complex, high protein content. Fine, with high protein content, and even if it is more nutritious than the human's milk. Okay, so it is having which protein? Lactoalbumin, right? Which protein was present in this milk, bache? Which protein was present here in this milk? It was alpha lactalbumin. It was alpha lactalbumin. Okay, so it is more nutritious than the human's milk for the babies. Okay, for the babies. Done, bache? So as I said, for the chemical safety testing, they are used. For even the vaccine safety testing, like polio vaccine is being tested on, right? Polio vaccine is being, right, it was being tested on transgenic mice. Right, it was being tested on transgenic mice. And then for the normal physiology and developmental study, I told you already, right? Insulin like growth factors for their studies, right? Even for the animals are used for investigating the lines of treatments for various diseases like cancer, cystic fibrosis, right? For cancer, cystic fibrosis, even for rheumatoid arthritis, which is autoimmune disease, you can note down Alzheimer's, right? So for such disease to study that, their line of growth, right, the transgenic animals are obtained. So MCQ can come from this part as well. This is important. Fine, bache, this is also important. Done, Rohini, Kevin? Rohini and Kevin are like, ma'am, we are not here to study, we are just here to pass our time, hai na? Okay, so that's what you need to study. So this is all about the transgenic animals, and transgenic microbes are also there, students. Transgenic microbes are also there. Like you have the example of Pseudomonas as well. Do you know the role of this transgenic microbe, especially the Pseudomonas? Anyone? Do you know the role of this transgenic microbe, especially the Pseudomonas? Yes, they are also the biochemical factories, right? They are also the biochemical factories. So Pseudomonas patida is used for alcohol fermentation. Imagine. It is used for what? Alcohol fermentation. So question can come from examples. So kindly note down. Okay, kindly note down. And this Pseudomonas is also used for, bache, removing the heavy metal pollution. Okay, it is also used for removing heavy metal pollution from water bodies, right? So Bacillus thuringiensis, it is used to produce the bio pesticides, right? So that is the example. But see, the way mice is also what? First of all, its lifespan also, right? And secondly, it is also the mammal, no? So the system somewhere is same. That's why the way. Only few changes are there, right? So next is, bache, the ethical issues, the biopatent, the biopiracy. These are the three important topics. Okay, so let's start with the ethical issues first. So what do you think about the ethical issues according to you? I'll take another 10 minutes and the class will be over. Fine. So when you talk about the ethical issue, so do you think it is ethical, or what do you think? Is it ethical or unethical? So Bhashini, Kevin, Govinda, what do you think? This biotechnology, this gene manipulation, is it ethical or unethical? But you see, there are both the things are there. No doubt, we are manipulating our crops, right? We are getting the better yield. Post-harvest losses are less. But if on another ground, if you will think, don't you think that we are playing with the biodiversity? We are promoting certain traits only, certain characters only, right? And obviously because of that, the another traits, the characters, they will be vanished, isn't it? They will be vanished, right? So of course ethical issues are there. Ethical issues are there. Ethically it is not right. We are, whatever is beneficial for us, we are focused on that only. We are propagating that animal or plants only, and we are even propagating that genes only. So obviously we are not, we are playing with the nature. So somewhere it is unethical, right? It is unethical, isn't it? We are just manipulating other organisms for our benefit. But still, right, let's say if there will be no restriction on all these processes, many scientists, you know that, right, it's all about the curiosity. It's all about the curiosity. So let's say if a scientist is very curious, like I want to try this, right? I want to make some transgenic animals that can fly, right? Yeah, transgenic, let's say human beings that can fly. Anything can happen. You know that the research on human is not allowed. But still, right, when it comes to the curiosity, we can do anything, we can make anything, and that can be disastrous also, right? It can result in the formation of a bio weapon, which are more and more dangerous. Do you know what are bio weapons? Like, imagine there is a disease producing virus. Let's say scientists they start creating disease producing virus, right? They can leave it. So obviously if they will pass that virus to another country, it can destroy the country. It can destroy the complete country. Bioweapons are that dangerous. Are you getting it? So obviously there should be some restriction on this type of research, right? There should be a committee which will verify, which will check that whether that research is for the well-being of the human being or the mankind, or is it affecting or not. So what we need to remember here, and that is the GEAC. What is it? GEAC. So question will come from this part, GEAC. It is Genetic Engineering Approval Committee, right? Genetic Engineering Approval Committee. Fine, bache, what is it? It is Genetic Engineering Approval Committee. So it was set up by the Ministry of Environment and Forest, that is MOEF. What is MOEF? You should know about these abbreviations, bache. Ministry of Environment and Forest. They have set up this, right? So Ministry of Environment and Forest. So they made this GEAC in India, which will, right, to regulate the research, testing, right, to regulate the research, testing, and even the release of the genetically modified crops, foods, and the organisms, right? Crops, foods, and the organisms. Clear, bache? Then comes the bio patent. Bache, whenever we talk about the patent, right, there is no need to make the notes as well. Everything is written in NCERT. As of now, you please understand this part, and then you have to read it from NCERT only. So patent is, let's say, if there is any discovery, right, there are three things that need to be considered: novelty, non-obviousness, and the utility. What need to be considered? Novelty, non-obviousness, and utility. Like, so if there is anything new that is discovered by a scientist, right, on what basis we are going to judge it? That invention should be new. When it is the novelty, means the innovation must be new, right? When it is the novelty, means innovation must be new. It should not be the part of any, pre-existing research or something, right? Or you can say that it should be completely new, right? First thing is this. Then non-obviousness. A new invention must produce unexpected or surprising result. It should not be like that someone has already, in their research they have mentioned that if we will do this, this can happen. No, it should not be like that, right? Means there should be a surprise element. Okay, non-obviousness. And then comes the utility, right? Then the last is what? Then the last is utility. The discovered fact or product should be a should be for the use of the human being, for a particular use of the human being. So these things are considered, right? What is considered? The novelty, it should be new, non-obviousness, like no one has predicted it, right? And the utility, right? On that basis, it is decided that this is a new discovery and we have to file a patent for this. So when we file that patent, no, so the government, the state government or the central government will allow us some special rights, right? Right, they will allow us what? They will allow us some special rights. So basically, when it is a patent, it is the official right from the government, right? Which is government. Then government will grant it to the inventor, okay, to the inventor. Clear, bache? So that only they can use it. Without their, without their approval, no one else can use it. They cannot sell it. So basically, that discoverer will have all the rights. Okay, okay. But when we have added the word biopatent, obviously then we are talking about the living organisms. Okay, so basically government, state government or central government will provide the special rights to the discoverer that this product belongs to you. If anyone wants to, sell that or want to commercialize that, they have to take the permission and they have to pay that person. Are you getting it? In simple language, in a layman language, I'm explaining you this topic. Okay, so when it is a living organism, then you are using the word biopatent. Okay, then you are using the word biopatent, right? So this is our right of that discoverer, right? One should know about it. If you have discovered something new, obviously you should apply for that patent and then you will get it. So this is about the biopatent, right, bache? This is about the biopatent. Done, bache? Done, done, done. But here we have one more example. Like we'll also talk about the biopiracy. Biopiracy, piracy, to steal something, right, without the knowledge of the owner, right? So if it is biopiracy, obviously you're talking about what? You're talking about the living things. Okay, you're talking about what? You're talking about the living things. So here we'll discuss about two things. There are two controversies in India regarding the patent and the biopiracy. Two controversies are there, and it is related to turmeric and neem. Neem, Edirecta indica, and the turmeric. It is related to that. Clear? It is related to that. Clear, bache? Clear, clear, clear. And moreover, even it is related to the rice as well. So rice, it is a very important food grain. Rice is our very important food grain. Yes or no? You know about it, na? And the presence of rice goes back thousands of years in Asia's agriculture history. When you're talking about the rice. So there are approximately 2 lakh, estimated 2 lakh varieties of rice in India alone. Just imagine. So diversity of rice in India is too much, right? Right? It is richest in the world. Clear, bache? It is richest in the world. And basmati rice is distinct for its unique aroma, for its flavor. So rice is there in Indian agricultural history from so many years. Okay, so many years. And I hope you know that 27 documented varieties of basmati rice are grown in India. You know, basmati rice, known for their aroma. So basmati, so 27 documented varieties. This is as per NCERT, write down. 27 documented varieties of rice, basmati rice, are reported in India itself. But here we have one story. So what happened in 1997, bache, American company, they got patent rights of basmati rice. Imagine, in 1997, American company, they got the patent rights for the basmati rice through the US Patent and Trademark Office. From US Patent and Trademark Office, they got the patent right. What they did, they just hybridized a semi-dwarf variety. What they did, bache Vishnu? What they did, they just hybridized, right? They just hybridized the Indian basmati with a semi-dwarf variety. Indian basmati with a semi-dwarf variety, and claimed it as a novelty, that it is something new, which is not, right? Which is not. Okay? Okay. Got it? Right? So it was unethical, na? Granting its patent to US was unethical. So it also comes under the biopiracy. Right? It can also be the biopiracy. It is unethical. And another controversy is regarding the turmeric. Just in May 1995, University of Mississippi, right? In May 1995, University of Mississippi Medical Center, right? They filed a patent for use of turmeric for bone healing. Imagine, Like they filed in 1995, they filed a patent for the use of turmeric in the wound healing. And you know that in India, if we are getting any injury or something, right, if we are getting any injury of something, you know that our parents are going to tell us one thing first, that we should take that, right, we should mix the turmeric in the milk and we should have that milk, right? The turmeric milk is always advised. So imagine they filed it and they got the patent as well. Okay, they got the patent as well. US Patent Office granted it to the University of Mississippi, right? Right. So obviously then it was challenged by Dr. R.A. Mashlekar, an Indian scientist. He challenged this patent. Okay, because he was aware. So awareness is important, bache. Awareness is important. Thank you, Vinod. Thank you so much for that praise, but it is not the fact. But still, thank you. Now please focus here. So it was challenged by Dr. R.K. Mashlekar, bache. He was an Indian scientist. He done a lot to awaken the Indian to intellectual property rights issues, and the same was done with the NEEM as well in 1996. Okay. Okay. So Vandana Shiva challenged the patent granted for fungicidal use of neem oil. Okay. 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 So biopiracy is theft or robbery of the biological and genetic resources, right? Right. Just say, let's say if there is a country, that country is a developing country, but another developed countries, they are using their resources. Okay, they are using their biological resources. So that will also, without their knowledge, without providing nothing to them. So that also comes under the biopiracy. Okay, that also comes under what? That also comes under the biopiracy. Getting it? Getting it, bache? So it is widespread in Asia and Africa, bache, where the most diverse ecosystem in world are found. Okay, so basically if I talk about this, so I can just give you one example of the brazine. It's a plant protein from African berries, right? I'll give you one example. It's a protein, brazine. It's a plant protein from African berries, but the patent was taken by the US. So again, it is also the biopiracy, right, bache? They got the patent, right? So they isolated that gene. The brazine has been patented in USA, but it is obtained from African berries, but it is patented in USA. They took that gene, they like synthesized it artificially as well and used it, which is unethical. Clear, bache? So Indian Parliament has recently cleared the amendment, second amendment, Indian patent bills, to take such issues into considerations. That's all, right? So as per second amendment bill, they have considered that issues into consideration. So this part is given in NCERT, so you have to read it from that from there, and it is right, you just need to understand the meanings. That's all. Clear, bache? So this is all about the biotechnology and its application. So tomorrow at 10 AM, right, we are going to meet on Unacademy platform. This first class is on I think now I should change the plan. Don't you think so? Okay, at 10 AM, let's practice biotech MCQs. What say? I have 2 slots for you. You will tell me, and accordingly I'll schedule my class. It is as per your comfort. So at 10 AM, earlier I was planning to take the class on urine formation. Fine, nephron and urine formation. And this at 5 PM, I'm planning to take biotechnology applications MCQ part. Now you tell me, what do you want? MCQ practice I was thinking to take. So you tell me, what do you want? It's okay, Soumyadeep. So I will just take your 2 hours. What do you want? You want this session at 8 AM, at 10 AM, or it's fine? It's fine. Okay, it's fine. So, okay, so one class is at 10 AM where we will discuss this topic, and one special class, right? It is at 5 PM where we are going to practice the MCQs of biotechnology and application. So, bache, these classes are free of cost. What you have to do, this is the coupon code that you have to use, okay? And you will be the part of my classes. So, go to the Unacademy platform, right? You can even search my name, just follow my profile, and you will get an idea of you will get the notification of my classes, okay? Okay, so which chapter you want to start next, do let me know, and see you all tomorrow at 10 AM on Unacademy platform. So take care. Thank you so much, everyone. Thank you.